Now we're going to study the use of microbes in the industry. Microorganisms are the organisms which are not seen with the naked eye and they survive as a single cell. They are in micron size and we cannot see them. So some probiotics or good bacteria, useful or microorganisms are bacteria, algae, fungi, protozoa bacteria, algae, fungi, protozoal virus. Cyanobacteria is used for the production of oxygen. Penicillium notatum is used for the production of antibiotics. And in the uses of microbes, we use microorganisms in food industry and medical field. In the food industry, for fermentation process, for the food additives, biofuel and biofertilization. In fermentation process, alcoholic, non-alcoholic. In non-alcoholic means preparation of beverages and dairy industries. In the medical, like milk, curd and cheese. In medical field, vaccine and antibiotics for the preparation of vaccine and antibiotics. Vitamin K supplements and fermented beverages also used in the yeast like beer, whiskey, lactobacillus for lactic acid, aspergillus niger for production of citric acid. These are the uses of microbes in industry. First is cyanobacteria is used for the production of oxygen and Penicillium notatum is used for the production of antibiotics. In the food industry for fermentation, alcoholic and non-alcoholic. In non-alcoholic for the preparation of beverages and dairy products, and in food industry they are used for the food additive preparation and also biofuel and biofertilization. Next, in the medical field, for the preparation of vaccines, antibiotics, vitamin K vaccines, antibiotics, vitamin K supplements, fermented beverages, lactobacillus used for the production of lactic acid, aspergillus niger used for the production of citric acid. So in the medical field, we use microbes for the production of antibiotics, vaccines, lactobacillus is used for the production of lactic acid, aspergillus niger is used for the production of citric acid, and vitamin K supplements and fermented beverages also. Next is production of enzymes. Using microorganisms, we can produce various enzymes like amylase, catalase, lipase, peroxidase, protease and penicillase. Now, enzyme. For the production of enzyme, we use sources like plant, animals and microorganisms. But microorganisms are mostly used because from this enzymes are easy to produce. Isolation and purification of microorganisms is easy and it is a cheaper source. Next is the production is more predictable. The production by using microorganisms is more predictable and controllable. It can also be easily synthesized. Next is physical parameters. So for the production of enzymes from microorganism, we need to maintain the pH temperature variation and we need to maintain physical parameters for smooth and quality production of enzymes. The pH temperature and aeration moisture should be maintained and physical parameters should be maintained for smooth and quality production of enzyme. Next is general production or general procedure involved in the production of enzyme. Selection of microorganism, media, substrate used, production process and isolation and purification. Selection of microorganism, media, substrate used, and the next is production process and then isolation and purification. Selection of microorganism, media, substrate used, production process and isolation and purification. These are the steps. First is a specific strain which acts as initiative molecule should be selected. In inoculum, a specific strain which acts as initiating molecule is selected. And the next is development of inoculum flask. With the help of media, we develop the inoculum flask. Then we introduce them into inoculation tank It is helpful for the stirring, produce aeration, then converted into transferred to fermentation tank for the stationary phase maintaining. Then cell lysis occur, filtration occurs, and then removal of nucleic acid occur. Then after removing of nucleic acid, salt treatment, then cold storage, then filtration and final purification. Common purification method is chromatography. After purification, freeze drying or lyophilization is there, then dry enzyme is produced. So this is the process, steps involved in the production of enzyme. Selection of microorganism, media, substrate used, production process, and then isolation and purification. The general procedures involved in the production of enzyme are: firstly, a specific strain which acts as initiating molecule is selected, and then the inoculum is developed in a flask using with the help of media. Then it is converted into inoculation tank. Stirring is there, which gives aeration, and then it is transferred to fermentation tank, where in the fermentation tank stationary phase is maintained, cell lysis occurs and filtration occurs. After filtration, nucleic acid is removed and salt treatment is done. After nucleic acid removal, after salt treatment, cold storage, and again filtration is done. After fermentation, cell lysis is done. After cell lysis, filtration. Between two filtration, nucleic acid is removed, salt treatment is done, and cold storage happens. Then after again filtration, final purification is done. Common purification method is chromatography. Then freeze drying or lyophilization is done, and then dry powder is produced. These are the steps. So after development of inoculum, it is transferred to inoculation tank, then transferred to fermentation tank, then cell lysis and filtration occurs. Between two filtration, removal of nucleic acid, salt treatment, cold storage occurs. Then after filtration, purification is done. Common method is chromatography. Then freeze drying or lyophilization and storage is done. These are the steps, general procedure involved in the production of microorganism.